Namaskar. Welcome to our channel Bio by Gulab Sir by Wisdom Coaching Center. As a part of series of lecture, today's topic is engineered insulin. As diabetes is the disease is because of lack of insulin that one has to suffer. One who lack the insulin and cannot control the sugar level in blood. So insulin is a natural product that was produced by the gene present in pancreatic gland, beta cells of islets of Langerhans. Artificially, to control this diabetic condition, it is a deformity, it is not a disease. It is because of lack of insulin, one cannot control glucose level. The very function of insulin is to convert that extra glucose into glycogen and stored into the liver, that you know very well. And to have that control of glucose, the diabetic person get administered by insulin from outside. That onset of diabetic person has to regularly take insulin through injections. Earlier it was so that insulin extracted from other organism or other body was being injected to control the diabetes. And it was the practice, but it was very costly. That extracted insulin was in very limited quantity, and moreover, second problem was having side effect. Generally, insulin is being extracted from the pancreas collected from slaughterhouses. These pancreas of pig, horses, cattle. When this insulin extracted from these animals, either it causes carrying allergy or some other allergic reactions or some other reactions, as this insulin, along with insulin, that other protein as an antigen from other animals would be transferring into the body of the person, causing some side effects. To overcome these side effects, there was an effort to have the production of insulin artificially. So, genetically engineered insulin is our topic. Genetically engineered insulin. So how we are lucky that to have now insulin in thousands of liters to administer lakhs of people. Earlier it was not so. It was very costly, or only few people, those who can afford that cost, could have getting administered. But now everybody can easily. It is the worth of DNA recombinant technology. But by the time when DNA recombinant technology was developed, that the gene which produces insulin having three major fragments or three major peptides in natural state, where to understand that, see, one, that A peptide this C peptide, and now this is B peptide. Now this A peptide produced from gene A, this is B peptide produced from gene B in a sequence, and join it with A peptide by this disulfide bond in natural state, and this is a C peptide. Now this is called proinsulin. This is called proinsulin. This is a structure of proinsulin. That means, another word if we have to use, prohormone. Prohormone. One more word we can use, proenzyme. Also we can use inactive enzyme. Why I use these three words? Because it is inactive in natural form. Suppose this gene A, B, C undergo transcription and translation, producing three peptide, and there is in our body is a natural method to have the removal of C peptide and have only N B. So that is called process, natural process, that to convert this pro-insulin into active insulin, that is removal of C peptide from peptide A and B and rejoining of this peptide A and B. So see this peptide A and another, this is peptide B, they are joined by this disulfide bond, and now this is a active peptide. This peptide A and this is peptide B are naturally joined in our body by this disulfide bond, and this is active insulin which work to control the glucose level in our body. So peptide A is made up of 20 amino acids. This is also very important point to note, that this peptide A is made up of 20 amino acids and peptide B is made up of 31 amino acids. 31 amino acids. So this is another point to be note for the NEET. Peptide chains are joined each other by disulfide bond. Problem was, we could got success to transfer the gene of insulin from human body to the bacteria to take the product, that is by DNA recombinant technology, and that we did understand in last lecture, that DNA recombinant technology to transfer the gene of our interest, desired gene, into the bacteria through Hector, that we learn, and to take the product from that bacteria. Now it is, when it is very clear to you to learn that that many scientists they tried and transferred this gene into the bacteria and they could produce these three peptides together, but they did not get success to have that process to separate, because bacteria as such is not a machinery to have that natural process of separation of C peptide from peptide A and B. So that was the problem. But in 1983, in 1983, an American company, Eli Lilly, see the company, name of the company, Eli Lilly, they have prepared two DNA fragment or two DNA sequences. They have prepared two DNA sequences, means two peptide chains separately, A and B, two DNA sequence similar to gene A and B separately. That means they have isolated gene A and B. They join that or ligated that gene A and B with plasmids separately in separate bacteria, could produce that peptide A and B separately and join artificially by disulfide bond to produce insulin from proinsulin. So production of peptide A and B was the success. Separate production of A and B was the success of Eli Lilly. Also, they got success to have the joining of that peptide separately produce A and B peptide. Suppose they have transferred that gene A and gene B to produce peptide A and peptide B, transferred by DNA recombinant technology. DNA recombinant technology into bacteria. Which bacteria they used? E. coli. Which bacteria they use? E. coli bacteria. And they produce peptide. They could extract. They could produce peptide. Peptide B and peptide A. Peptide A and peptide B separately, isolated from that bacterial culture, and now these two together that peptide A and peptide B together. Peptide A and peptide B together. They could got success to join it by disulfide bond, and that was the greatest achievement from their side to produce the same enzyme which naturally produce in our body. They could produce artificially the same enzyme. Why I use word enzyme? Because I use word proenzyme. Same hormone, same insulin which produce naturally in our body. See, this is the insulin which naturally produce in our body. Now this is produced by Eli and Lilly company artificially by transferring gene A and B separately into bacteria to produce peptide A and B, and later that peptide A and B, see, peptide A and peptide B, these are produced artificially and joined by disulfide bond. Thus, they got that success to produce insulin from proinsulin. Proinsulin is an inactive form. Insulin is active form. That they did separated this gene A and gene B from bacteria. That is what prepare two DNA sequences similar to gene A and B. That means isolated from bacteria. That gene, they transfer that gene by DNA recombinant technology through plasmid into the bacteria. There they could express and produce two peptide A and B. Then that peptide A and B they brought together and joined it by disulfide bond to have the similar structure to the naturally produced insulin. And thereby now this is being a artificially produced, genetically engineered. This is being genetically engineered, that is artificially produced insulin is finally come into the market, also called as humulin. This human insulin also called as humulin. So it is famous by our other name humulin. So here we did understand how this Eli and Lilly company they could get success to produce this insulin artificially in bacteria by DNA recombinant technology. As we know that bacteria has very high rate of production, they get double within 20 minutes. So as many as bacteria produce, that many copies of that gene also produce, and in bioreactor, in this bacteria being cultured, with that cultured bacteria undergo gene in bacteria undergo transcription and translation and keep on producing this peptide A and B continuously, so that Eli Lilly company could produce thousands of liters of insulin per day. Now all the companies under property act or royalty under patent of Eli Lilly company, hundreds of companies in the world, they are continuously producing this insulin and marketing it. So lakhs and lakhs diabetic persons are being administered, they are blessed, and this is really very worthwhile or worth work is being carried out by Eli Lilly company. So it is the boon to the diabetic person that any quantity of insulin is available to the in the market to administer lakhs and lakhs diabetic person. So today we did learn that how this genetically engineered insulin is being produced, and now today it is marketed. So I hope you did understand that how this insulin is being artificially produced in laboratory. It is a matter of pride for us that we scientists, we biologists, that working day and night for the well-being of humankind through various areas. So this is one of the area that production of insulin on artificial level is boon to the humankind. So I hope you did understand what I did explain today's lecture. This question can be asked, write short note on insulin produced by Eli Lilly company, or explain how that production of insulin on artificial level is carried out, or explain the process of production of insulin on artificial level, artificial process developed by Eli Lilly company. Why that natural insulin which extracted from other animal not found sufficient enough and effective enough to treat the diabetic person? This question also can be asked. In NEET question can be asked, one of the following company has developed insulin on artificial level is. Also can be asked how many amino acids are engaged in peptide A and peptide B. So 20 amino acid in peptide A and 31 amino acid in peptide B. So proinsulin converted into insulin is the success that Eli Lilly company has got. Therefore, we have now thousands of liters of insulin to administer the lakhs of diabetic person. If still you are in doubt, you can ask in comment section, and I appeal you to like our channel by subscribing it and by pressing bell icon to get notified time to time whenever new videos are uploaded. Thank you.